Welcome to our first video in our series on cellular respiration, which simply put is the series of chemical reactions that are used by a cell in order to produce ATP. Now, the reason why we spend an entire unit basically talking about cellular respiration is because of the sheer universality of several processes that are all included within the term cellular respiration. Because every organism requires ATP, basically every organism, including plants that make their own glucose, actually uses some form of cellular respiration. So this introduction is meant to give students a baseline about the general processes that are used in order to convert a molecule like glucose into ATP. Now, technically, cellular respiration can use any sugar molecule as long as you have the enzymes to break it down, or even other molecules such as fatty acids that are also energetically rich, but because of the fact that glucose is the most common energy source used by every organism on the planet, we're going to be using this as a baseline. So you might familiarize yourself, or you might already be familiar with this basic chemical reaction here, where we take the chemical formula for glucose, C6H12O6, and we know that glucose can only react when it's in the presence of oxygen gas, and this is literally the primary reason why we need oxygen gas in the human body. And we can recognize the breakdown of glucose as a combustion reaction, meaning the two compounds that we would get would be carbon dioxide, which we would exhale via our respiratory system, and water vapor, which again would become part of the 70% of our bodies that is composed of water. But of course, what we are interested in by doing this reaction is the energy that we get as an output from this reaction, which we would then convert into ATP. And the process of how we do that is the focus of what we're going to be learning about in this series of videos. So why glucose? Well, as we can see from the line structure of glucose is that glucose has lots of bonds, and as we know when we talked about what ATP actually does, the more bonds a substance has, the more potential energy is stored within the bonds of that molecule, because the electrons within those bonds are screaming out, not literally of course, to be released and converted into a usable form, which we call ATP. So more specifically, we can say that glucose has a large amount of high potential energy bonds, meaning they are bonds that generally want to be broken. Now, this might cause some confusion in some people, because if glucose is so unstable, naturally this chemical reaction would happen spontaneously, but when was the last time that you saw a glucose molecule catch fire by itself? Now, chances are the answer to that is never, because glucose doesn't do this. And it doesn't do this because of a concept called activation energy, meaning that it doesn't do this because in order for glucose to be broken down, a certain amount of energy needs to be entered into glucose in order to make it more unstable so that it would spontaneously break down. Now, the other reason why glucose doesn't do this in the body is because of the fact that cellular respiration is an exothermic reaction, meaning that it produces a large amount of energy, and that's exactly the point. We want this energy to be captured in the form of ATP, but if glucose were simply to catch fire and burn by itself, all of this energy would be wasted and would be released into the surroundings as heat, which is not very useful for the purpose of our body. The other major problem of doing this is that this will massively increase the temperature of the body, and any significant temperature increase is going to cause massive problems involving protein denaturation, including all of the enzymes that catalyze cellular respiration reactions. So as a consequence of this, we cannot break down glucose in one single massive energy releasing step, but instead need to break that process down into 3 sequences of chemical reactions that make up the 3 components of the cellular respiration unit. The first of which is the truly universal process that all living things share, is the process of glycolysis that simply occurs as soon as glucose enters the cytoplasm of a cell. And this is why this process is done not just by eukaryotic cells like human cells, but also in the simplest forms of bacteria as well. However, eukaryotic cells have specialized organelles that we call mitochondria that are specialized in order to produce ATP in much greater quantities, and the first sequence of chemical reactions that occur in cellular mitochondria are what we call the Krebs cycle, although more technically known as the citric acid cycle, referring to the central compound in the cycle itself. Finally, the components produced in the Krebs cycle are used in a process that we call the electron transport chain, which is abbreviated as the ETC for short, which also relies on using the cell's mitochondria and specifically relies on the distinctive double-layered structure of mitochondrial membranes in order to function. However, you can consider this all of a precursor, as we will be taking a look at each individual sequence of reactions in their own multi-part video series. To begin, if we don't have glucose inside of a cell, none of this actually matters because it all hinges on breaking down glucose. So to start, if we remember our membrane transport systems, we can recognize that glucose is definitely too big in order to pass through the individual phospholipids of the phospholipid bilayer, and we also recognize that it is too polar to be able to move through the nonpolar sections of the phospholipid bilayer, which means that glucose, like every other large polar molecule, requires a specific carrier protein with an active site that is complementary to glucose's structure in order to enter the cell. And as long as we have a higher concentration of glucose outside the cell than we do inside the cell, glucose will always be able to diffuse in. But the problem is, number one, this diffusion is not going to last forever, because as this concentration outside decreases and this concentration inside increases, this is going to result in an isotonic situation where glucose actually needs to be pumped into the cell, which wastes energy, and that's something that we do not want to do because we always want to be using glucose to produce energy. We don't want to spend energy in order to make energy. So therefore, we need to keep this concentration of glucose in the cell low in order to make sure that the concentration gradient allows glucose to flow one way. Now, is it possible to keep this concentration lower than the concentration outside all of the time? Obviously, the answer is no. So if the inside of the cell has a higher concentration of glucose than the outside of the cell, because carrier proteins are bidirectional, this means that glucose will want to diffuse outside, and this is something that we never want to happen. Therefore, we need a way to stop glucose from diffusing out of the cell. And the first process of stopping glucose from diffusing out of the cell is the very first step of cellular respiration, which is where our next video is going to pick up from.